So in this lesson, you're going to learn how to outline the benefits of using databases that provide information about nucleotide sequences of genes and genomes, and amino acid sequences of proteins, as well as protein structures. So what does it mean? So we know that many data means results, right? Results from experiment, means research. Right? Data means results, isn't it? From experiments of research. So there are many data that have been obtained from scientific research in the world, because every day there are so many scientists in many universities and research labs that are performing research, and many data have been produced daily and published daily and presented. So what are the data so far we want to know that have been obtained from the experiment and research? A, the DNA-based sequence of human genome. So this one complete already, this project already completed. We already know the DNA-based sequence of human genome through Human Genome Project. And from there, they also found out the base sequence of human genes. And there are scientists that also study gene expression, right? So whether yes is expressed, no is not expressed, and degree of expression, in which cell is being expressed, and when it's being expressed, especially during development of embryo, for example. In which cell is being expressed means whether a normal cell or the disease cell, for example. We also got scientists that study protein structure. So from there we know we have data about the primary structure of protein. Primary structure means amino acid sequence. Then we also have scientists that study the tertiary structure of protein, means the 3D shape of the protein molecule. And then they also learn the function of the protein. So these are examples of data that have been available due to scientific research. So the question now is, these data are stored in digital form in a variety of databases. Let's say, for example, you are from Taylor's College. So maybe Taylor's College got a cloud system, right, an online storage system where all the lecturers and professors and also the students, when they've done research, they got their findings, they presented paper. So they got this data that are all being stored in databases of Taylor's College or Taylor's University. It's only accessible for the staff there and students there only. So these data are stored in digital form in a variety of databases everywhere in the world. So the question here we want to find out is how to develop a software system in order to store those data, and then to go and search those data and try to retrieve those data and try to analyze this data. So this is the main topic of this lesson. Can there be a system that we can develop? And usually who will develop this system is actually the programmer, because they need to develop the software so that there's this programming that instruct the computer. So now when you got computer science, you got this software as well as the hardware, right? So hardwares. So these two are part, if I'm not wrong, under computer technology. Computer technology. So computer technology will now come in here so that we can develop software, okay, and hardware that can help us to store the information, search the information, retrieve the information, as well as analyze this information, so that other scientists can use this data to build new knowledge for their research. So I will give you an example. What does it mean? So this is part under the science called bioinformatics. So bioinformatics is biological studies that involve collection, processing, and analyzing of large amount of biological information and data. Actually, this biological information is data, right? In the databases using computer software. So this field is packed under this big title called bioinformatics. So the scientists want to be able to store the data, collect the data, and then process means analyze it to obtain new knowledge out of it, to help them to design a new research to find out new findings. For example, let's say COVID. So after the COVID starts to spread all over the world, so what do the researcher do? They find out what is the source that caused the disease. So they find out it's the virus. Then they go and find out, so what is the base sequence of the DNA of this virus? Okay, then from there, they go and find out for this base sequence, what is the antigen of this virus? Okay, then after that, they're going to find, or maybe RNA of this virus, and going to find out, okay, so what antigen I can introduce to human body to act as an antigen in the vaccine to vaccinate this person to stimulate immune response so that memory cells are being produced. So can you see? These are application of all the basic knowledge. So the scientists all over the world are doing research, and whatever finding they have, if it's made available, then the scientists now can build on the knowledge. They don't have to start from scratch. So this is called sharing of the data to help develop a vaccine that is effective against the virus. And of course, they've got scientists that actually try out the vaccine and see how well it performs, right? Whether there's any side effects or not, and how long it can perform. So these are all results of research of many scientists put together to benefit us. So now when we step back and look at it, whatever that we receive now, right, is actually the effort, the knowledge from so many scientists and the time that they've put in to create something that is useful for us, so that now, because we are being vaccinated, we are no longer so scared about actually contracting the COVID, because we can recover very quickly because we've got memory cells against them. So can you see how the little contribution of the scientists in every field, when you bring together, it actually benefits us. So that is what we want to do when we study the basic and applied knowledge in the research to obtain the results. So that's how everything put together and how it works in science, right? So now you're learning small things. Don't look down on these small things. It actually can create miracles. It can create miracles if you learned it well and you apply it well. Okay. So in short, bioinformatics involved biological data that's been obtained, computer technology that work on the software and the hardware, as well as statistics, means mathematical methods. So you know, statistical analysis means you use statistics to compare two sets of data, and to be able to compare two sets of data or three sets of data or many sets of data. So this is called statistics. And then to see which one is more effective, which one is less effective. If you try out drug A, B, C, D, E, right? Then to compare all these drugs, how effective it is, which vaccine is more effective. So you have to use statistics, right? After so many people vaccinated, how many people are immune for very long? How many people that the memory cells still remain for very long in the body? So this one you have to use statistics to compare among different vaccines, for example. This is called statistics. Okay. So this is bioinformatics. This whole field is called bioinformatics. So putting it in a pictorial form, these are all the research that has been done by the scientists, and a lot of different informations have been obtained. The one that you need to learn from your syllabus is only the DNA-based sequences, gene-based sequences, protein primary structure and tertiary structure only. But other than that, and also protein function, other than that, there are many other information that's obtained from the research. Okay, that's shown in this diagram. You don't have to know, this is just for you to see only. Okay, so I have the data now. Now what? So all this data available. So now what? Now we use bioinformatics to store the data, to search the database, to retrieve the data that we need, and then to use software to do analysis that we need to analyze to obtain biological insights. Oh, vaccine A is more effective than vaccine B. Oh, vaccine A got side effect. Vaccine B got less side effect. So this is called biological insights. So these are examples of databases, right? Database ensemble and database uniport. So database ensemble hold data of genomes of eukaryotic organisms, which include genomes of human, zebrafish, mice, because these are organisms that are highly used as research organisms. So therefore, scientists have found out their genomes. And then database uniprot, prot means protein, universal proteins, so whole data of amino acid sequence and function of proteins. So these are examples of the databases. Then example of search tool so that you can go and access this database and search out the information that you want. Just like Google, you key in a keyword, they try to search in their database and present to you, oh, these are the information that matches your keywords that's available in my database. So this is search tool or search engine that's available. Example, BLAST. Okay, BLAST is an algorithm, is a procedure for comparing primary biological sequence information. Example, comparing base sequences of different genes. So you've got this software for you to compare base sequences of different genes or compare amino acid sequence of different proteins, for example. Then, so after this bioinformatics system has been developed, what it can be used for. So these are examples of the use of this bioinformatics, because you can use to store, to retrieve the data, to compare the data, to analyze the data. So these are examples of what it has been used for. So number one is to analyze the DNA base sequence and protein amino acid sequence. So what is the base sequence of this gene? What is the base sequence of the COVID virus? Or what is the RNA sequence of the COVID virus? What is the protein amino acid sequence of the antigen of the virus? Number two, aligning different DNA and protein sequence to compare them. So we want to compare between the DNA base sequence and the amino acid sequence of the protein, whether this protein is actually coded by this piece of DNA, right? For this protein that can be found in this virus, which particular gene code for this protein? So we have to compare the base sequence of DNA to the amino acid sequence to match which gene code for this protein. Number three, creating and viewing three-dimensional models of protein structure. So after I know the amino acid sequence, right, I can use the software to create a three-dimensional structure of this protein and play with this protein. Why they want to play with this protein? They want to find out. Let's say I want to look for drugs that can inhibit this protein. So you know the inhibitor have to bind to active site or allosteric site, right? So once I create this 3D protein, and I've got this drug 1, drug 1, 2, 3, 4, 5, I want to know which drug that I've created in the lab is able to bind to this protein and therefore inhibit its action. I can use the software to actually try it out and compare rather than try out on organism. I can do it straight away in my computer screen. Isn't it better, faster, cheaper? Next, extracting patterns from large data sets in order to compare them. So are there any other researchers that have done a similar experiment? So can I go to that database and extract out other possibilities? Let's say I've researched drug 1, 2, 3, 4, 5. And another university, they have actually researched drug 7, drug A, B, C, D, E. So can I extract them and compare them and see which one more effective? Right? When they say more effective in their research and my more effective in my research, it can be very different. So I must bring everything out and try to fit that drug into the protein active site or allosteric site and see which one fit the best. Correct or not? Now I can compare among all the choices I have, which one is the best. So I can do that. So compare between human genome and genome of other organisms. So if you want to see genetic relatedness between human and other organisms, we can compare. Comparing the analogs. So this one I mentioned just now. Compare analogs of protein as possible drug that can bind to pathogen enzyme to inhibit it so that it will not cause disease. So this one I've just mentioned just now. Okay. Right. So here, so these are examples of comparing human genomes with genome of other organisms to get some information out of it. So why they want to compare human genome with this one is Drosophila fly. Now, Drosophila is actually the fruit fly. You know, when you cut fruits and you leave it there for some time, tiny little flies will start to come and visit the fruits. So these are Drosophila. They are highly used as research organism in the lab because they can grow, they can multiply very fast. So it's very easy to grow them, to use them to do research. So scientists have compared the genome of human with the genome of the fruit fly. Fruits, F-R-U-I-T, fruit fly. Fruit fly, Drosophila. Species name, Drosophila. Species name, I need to underline this because it's species name, okay? Because I cannot italize it. So to underline species name. See, compare. And can compare it with the genome of nematode worm. So this is nematode worm here. So compare with this genome of the worm. And so compare it with the genome of the malaria parasite, which is Plasmodium. Right? So why they want to compare? Because they want to find out certain information and see whether it matches or not. Because we cannot grow human and compare them, right? We don't take human in the lab and try to grow them and try to do research on them. So because of that, we use alternative. We use fruit fly, we use nematode worm, we use malaria parasite, and we use them in the lab and we grow them and then we test them and then compare them. For example, if we have the gene and the fruit fly also got the gene, how the gene, what does the gene code for in the fruit fly? When it's being expressed? So does it work in the same way in our body during, for example, development? So we want to compare how our human development occurs from zygote to embryo to fetus. And how does it development occur in the fruit fly? Can I use fruit fly as an example and see the genes that switch on during development and then see, do we have a similar gene? So this is how the genes being switched on in human during development. So we can do that. Okay, so when the genes are being switched on, what is the protein that's being produced? What is the amino acid sequence of the protein? What is the three-dimensional structure of the protein? What is the function of the protein? So we can actually do research based on that one. Same as for nematode worm. And then for malaria parasite, what we want to find out is that, so what is the gene that code for the antigen for the malaria? What is the base sequence? So that I can synthesize the antigen and use it as a vaccine against infection by malaria. So this is a very powerful tool. Okay, so put all in all recap, bioinformatics is a biological study that involves the collection of all the biological data obtained from biological research, process it, and analyze this biological information and data using computer software to derive to new knowledge, new insights. So this involves multidiscipline, like you can see here, involve biology, medicine, databases, text mining, web application, algorithm programming, statistics, mathematics. So many disciplines will be involved in bioinformatics. So you have learned in this topic to outline the benefits of using databases that provide information about nucleotide sequences of gene and genomes, amino acid sequences of proteins and protein structure. So what I want you to do is you try out this past year question, which is really, really good. This example will show you how we can apply this bioinformatics to obtain new information. This is a very good past year question. I've put it into the PowerPoint. Okay, so therefore, please do them. So at least you understand what is bioinformatics. So this is only one example, but I think this is very good. So this is your homework. Go through this past year question and do the question and read the answers and try to understand what is bioinformatics. All right, so that's all for today's lesson. Okay.